Here we're going to talk about the mechanisms for regulating gene expression, which genes are activated and being actively transcribed into RNA and then translated into proteins. So gene expression is the process of making proteins based on the information in the DNA. So this is what we call the central dogma, where we have the DNA in the nucleus of the cell, either prokaryotes or eukaryotes. That DNA, it's transcribed into RNA. RNA brings that information to the ribosome, which then translates the RNA into protein. So we call transcription when we go from DNA to RNA, because we really haven't changed the language. We're still using nucleic acids, which is changing a DNA sequence, and we are transcribing that into an RNA sequence. It's called transcribe, just as you would take from your textbook that is printed letters into handwritten, but you're still using the same language. Translation now is a different language, because now we go from nucleic acids, from RNA, into proteins. So now the sequence of RNA is now being translated into a sequence of amino acids. So now we're using a different type of macromolecule. So going back to the central dogma, this is eukaryote that has a nucleus. The process has some additional steps. So we start with transcription, where we make an RNA copy from the DNA information. That RNA in eukaryotes has some modifications, that is post-transcription. So after it's been already transcribed into RNA, that RNA gets modified. Then that RNA exits the nucleus and starts being translated in the ribosomes, which are now located in the cytoplasm. Once we have the protein, the polypeptide, that polypeptide goes through folding and then post-translation regulation. So we can deactivate or activate the protein after it's been made. So all those steps are steps in which we can regulate gene expression. So we can regulate whether RNA gets made, how it gets transformed once it's made, which is post-transcription, whether it's actually translated at the ribosome, and once the protein is made, we can also modify that protein. In prokaryotes, since they don't have the nucleus and translation happens right as transcription is still going on, that leaves no room for post-transcription regulation. They go from transcription, we can either prevent transcription, the RNA from being made, but once the RNA is there, it goes into translation right away. So we miss those steps in prokaryotes. So let's look at the first step in regulation and transcription, and this exists in both prokaryotes and eukaryotes, so whether we can use that DNA as a template to make an RNA copy. This is all based in proteins that bind to the DNA, so we call those DNA binding proteins, and those are proteins that have a DNA binding motif or binding domain, so sort of repeated shapes that proteins have that allow them to bind to the DNA. An example of this type of regulation, for instance, in prokaryotes, we have the lac operon, where you have a protein called the lac repressor that binds to the DNA and creates a roadblock so that RNA polymerase cannot transcribe the genes that are encoding for enzymes to digest lactose. If there is no lactose present, the lac operon will bind to that DNA region and prevent the transcription of lactose-related genes. In eukaryotes, we also have transcription regulation, but here's a little bit more complex. If you remember, transcription in eukaryotes requires multiple factors, and the promoters can be located far away from the actual coding region. So look at transcription regulation in eukaryotes. As we said here, the promoter region can be farther apart from the coding region, or the region that will be actually transcribed. Some proteins can bind far away from when we're about to transcribe and either promote or repress the transcription of the gene. Even if proteins are going to promote transcription, you still need a large combination of different proteins. So in eukaryotes, you need multiple transcription factors in order for transcription to start, in order to recruit or bring over RNA polymerase. So all those transcription factors, the right side of them needs to be in place so that RNA polymerase can come in and start transcribing the gene. There's an additional way in which eukaryotes can control transcription, and it is because in eukaryotes, the DNA is tightly packed around histone proteins, and how tightly that packing is can interfere with transcription. So if you think DNA is too condensed or too packed, it will be very difficult for RNA polymerase to come in and transcribe that DNA. So packing the DNA closely inhibits transcription in eukaryotes, versus making it more loose will make transcription of those genes more likely. In eukaryotes, you can regulate the likelihood of transcription by packing the DNA more tightly, and one way of doing that is packing around the histones. You can also add methyl groups to the DNA that would deter RNA polymerase from coming in, or you can make it more loose and more likely to be transcribed by adding acetyl groups to the histones, and this will kind of make them spread out and loosen up the DNA so it's more available for transcription. So this is unique to eukaryotes. There's also another step that is unique in eukaryotes, it's not present in prokaryotes, and it's the time that happens between the RNA being transcribed and the point where it goes to the ribosome. So in eukaryotes, since transcription happens in the nucleus, before the RNA exits the nucleus, it goes through a series of post-transcription modifications. And those post-transcription modifications give us an opportunity to again regulate gene expression. For example, in eukaryotes, the RNA gets spliced. In eukaryotes, RNA has exons and introns. Exons are the ones that actually code for information, and introns get removed. And you can have different combinations of exons and introns in the same RNA transcript. So that by creating multiple combinations, you can have multiple proteins made from the same DNA sequence. This is what is called alternative splicing. So you can produce multiple messenger RNA from the same RNA transcript just by removing different pieces of it. So just think of it as like creating a different puzzle from the same RNA transcript. Another thing that happens in eukaryotes, since the RNA transcript has to exit the nucleus and go into the cytoplasm, you also do some modifications to help that RNA leave the nucleus and also to keep it stable while it is in the cytoplasm and before it gets translated. Those modifications is adding a 5' G cap, guanine, and at the 3' end you add a poly-A tail, several adenines to the 3' end. And this helps keep that messenger RNA stable, prevent it from degradation so that it can be translated. Again, this doesn't exist in prokaryotes because in prokaryotes, translation follows right after transcription. The next step is once the RNA exits the nucleus and is getting ready for translation, here in eukaryotes, you still have an opportunity to do more gene regulation. So even at this point, you can still stop the process or change it. One way in which this happens is once the RNA exits the nucleus, it can be prevented from being translated by joining other pieces of RNA that are complementary to the messenger RNA. This is what we call RNA interference. This is when complementary short pieces of RNA bind to the messenger RNA and target that RNA for destruction. So even though we already had the RNA transcribed and we already modified it and it already exited the nucleus, you can still stop that process in eukaryotes by using RNA interference. Another form in which you can control gene expression. So here we already had transcription, we had post-transcription regulation where we modified that RNA exited the nucleus and it was translated. So the protein was made. We can still modify that protein, which is called post-transcription regulation, to change the phenotype of that cell. One way could be by destroying that protein as soon as it's being made or quickly after it's been made. This way, the protein only acts for a very short period of time and we can quickly get rid of it. The way the cells do this is by using a tag, sort of telling this protein should be destroyed. That tag is called Ubiquitin, which is another short peptide that is added to proteins, and you add multiple of those and it acts as a signal to get that protein destroyed. Proteins that are targeted with Ubiquitin get degraded in the proteasome. The proteasome is a sort of organelle that destroys proteins. It's just a cycle of Ubiquitin. You add Ubiquitin tags to the protein that you want to destroy. Then that protein gets into the proteasome that breaks it into small polypeptide fragments, and the amino acids can be recycled. Same with the Ubiquitin pieces can also be recycled. So just to summarize how we can regulate gene expression, in prokaryotes we can regulate gene expression before the process gets started, so preventing or promoting transcription, which will be transcription regulation, either promoting or increasing preventing RNA polymerase from transcribing the DNA into RNA. Or at the end of the process, which is post-translation, once the protein is made, we can just target that protein for destruction. Eukaryotes have a little bit more room for regulation, so they can also do transcription regulation, either prevent or promote RNA from transcribing DNA into RNA. Post-transcription regulation, which is since eukaryotes have multiple steps between transcription and the RNA getting translated, we can have different regulatory mechanisms in between. So for example, you can have different splicing of the RNA. You can add the poly A tail and the 5' G cap to help protect that messenger RNA, or you destroyed it or not let it exit the nucleus. All these are ways of post-transcription regulation. So this is what happens between the RNA transcript being made and it getting to the cytoplasm for translation. Since translation doesn't happen right away, you can also prevent the RNA from being translated. Once it is in the cytoplasm, you can block it with RNA interference so that it doesn't get translated. And the last one, which is also present in prokaryotes, is even once you make the protein, you can still deactivate that protein, either by, for example, phosphorylating or dysphosphorylating is a way of post-translation regulation. But you can also break down that protein by targeting it with ubiquitin and then getting that protein destroyed. I also forgot to mention in the first transcription regulation in eukaryotes, it can also happen not just by having DNA binding proteins that increase or decrease transcription, but eukaryotes also have another way of doing it, which is by packing that DNA tightly or letting it loose, making it more available for transcription.